Human development begins at conception, the exact moment a single sperm penetrates an ovum. The merging of their genetic material creates a zygote. It is a single cell measuring just 1/20th the size of a pinhead, yet it contains a complete set of 46 chromosomes, derived equally from both parents. Inside the nucleus of this zygote is deoxyribonucleic acid, or DNA. It forms a double helix structure that acts as a chemical blueprint, tightly coiled into those 23 chromosomal pairs. Before a cell can divide, each chromosome must duplicate itself, resulting in this characteristic X shape. The original strand and its copy are bound together by a central structure called a centromere. Extending outward from this point are the short and long chromosomal arms. The zygote holds the entire instruction manual for a human being, but scaling that single cell into a mature organism with trillions of cells requires two highly specialized engines of replication. The first engine is mitosis, the process responsible for everyday somatic growth and tissue repair throughout our lives. During mitosis, a cell duplicates its 46 chromosomes and then divides exactly once. The result is two genetically identical daughter cells, each retaining the precise 46 chromosome blueprint of the parent. The second engine, meiosis, serves a strictly distinct purpose. It operates exclusively in the testes and ovaries to produce gametes, the sperm and egg cells. In meiosis, the parent cell also duplicates its chromosomes, but instead of dividing once, it undergoes a sequence of two separate divisions. This double division produces 4 distinct gamete cells. Each one contains exactly 23 single unpaired chromosomes, exactly half the genetic material of the parent. Mitosis preserves the genetic code with exact fidelity to build the body. Meiosis intentionally halves the code to prepare for the creation of a new, unique organism. Meiosis generates genetic diversity through a process called crossing over. This diagram shows crossing over in action. After duplication, maternal and paternal chromosomes align. Their arms cross over and break, forming a chiasma. The broken segments swap places, yielding gametes with entirely new hereditary combinations. This process introduces a specific mechanical vulnerability, non-disjunction, where chromosomes fail to separate properly during the final split. The chiasmata formed during crossing over are required to tether homologous chromosomes together for proper segregation. If no chiasma forms, the pairs drift independently, leading to the catastrophic chromosomal imbalances seen in non-disjunction. When non-disjunction occurs, the resulting gametes end up with an irregular number of chromosomes, a condition called aneuploidy. Looking at a standard karyotype, the most common autosomal aneuploidy occurs here, at the 21st pair. If the zygote inherits an extra copy of this chromosome, it results in trisomy 21, commonly known as Down syndrome. This extra genetic material produces a distinct phenotype, characterized by a round face, flattened skull, and specific intellectual and motor delays. Clinical data shows a strong correlation between these non-disjunction events and advanced maternal age. Over decades, the meiotic machinery in the ovaries accumulates the effects of environmental insults. As the cellular mechanisms become less efficient, the likelihood of a segregation error on the 21st chromosome steadily increases. The failure of a single chromosome pair to detach during meiosis results in a child with a specific, lifelong set of physical and cognitive differences. Beyond the 22 pairs of autosomes, we find the 23rd pair, the sex chromosomes. These dictate male or female development, and errors here produce highly specific outcomes. For instance, if a female inherits only a single X chromosome instead of 2, the resulting genotype, XO, causes Turner syndrome. Females with Turner syndrome typically exhibit short stature, a webbed neck, and infertility. Interestingly, while spatial reasoning might be affected, their verbal intelligence usually remains completely intact. Conversely, a non-disjunction error can result in a male inheriting an extra X chromosome, producing the XXY genotype of Klinefelter syndrome. This extra chromosome disrupts normal male development, leading to undeveloped testes, enlarged breasts, and measurable impairments in motor and language abilities. Not all chromosomal anomalies involve a missing or extra structure. Fragile X syndrome occurs when a specific section of the X chromosome becomes tightly constricted and physically breaks apart. Because males possess only one X chromosome, they lack a second, healthy copy to compensate for the damaged genes. As a result, they experience more severe cognitive deficits and learning disabilities than females with the same mutation. Errors on the 23rd pair isolate their damage to sexual maturation and specific cognitive domains. They demonstrate that the sex chromosomes operate with precise, targeted influence over human development. Downward from the macroscopic scale of entire chromosomes, developmental anomalies also stem from microscopic errors in specific genes, known as alleles. Phenylketonuria, or PKU, is a recessive genetic disorder. A microscopic coding error leaves the individual unable to produce the enzyme required to metabolize the amino acid phenylalanine. PKU demonstrates that a genetic defect does not inevitably lead to a disorder. Because the accumulation of phenylpyruvic acid can be prevented by replaced nutrients, the same genotype results in different phenotypes depending on the nutritional environment. Other recessive genes express themselves through codominance, where the mutation directly alters physical structures. A primary example is sickle cell anemia. This micrograph contrasts normal red blood cells with sickled cells. The recessive gene forces the typically round cells into an elongated, hook-like shape that limits oxygen transport and clumps in the circulatory system. Some micro-level mutations are far more devastating. Tay-Sachs disease is a recessive disorder that prevents the breakdown of lipids in the central nervous system, leading to toxic accumulation and inevitable early death. Meiotic errors change the broad architecture of the body. Gene-linked mutations operate as microscopic typos, silently dismantling the highly specific biochemical pathways required to sustain life. Even if the genetic code is assembled flawlessly, the physical mechanics of gestation present their own vulnerabilities. This is most critical between weeks 3 and 8, the embryonic stage. During this window, the embryo's inner mass differentiates into 3 distinct layers. The outermost layer, the ectoderm, is tasked with forming the central nervous system. Around the 4th week of gestation, a section of the ectoderm must physically fold inward upon itself and close completely to form the neural tube. If the spatial mechanics of this folding fail, the tube remains open, resulting in a condition called spina bifida. Clinically, this means the spinal cord and its protective membranes physically protrude from the infant's spinal column, causing severe nerve damage and motor impairment. Genetics provides the blueprint, but embryogenesis requires physical execution. A failure in the structural geometry of tissue formation is just as critical as an error in the DNA. To understand these pathologies, we map them against prenatal chronology. Macroscopic chromosomal errors, like trisomy 21, originate during meiosis, locked in at conception. In contrast, structural defects like spina bifida appear weeks later, triggered by mechanical failures during the embryonic stage. Diagnosing a developmental failure requires identifying both the biological system that collapsed and the exact moment on the timeline when the failure occurred. Today, the clinical reality of pregnancy involves actively monitoring this perilous timeline. Mastering the mechanics of heredity enables modern interventions. Through genetic counseling and procedures like amniocentesis, we can identify meiotic errors and gene-linked mutations long before birth. Understanding the specific mechanisms of a disorder allows us to intercept it. We can implement a precise diet to neutralize PKU or administer hormone therapies to mitigate the physical effects of Turner syndrome. By rigorously mapping the mechanical vulnerabilities of human development, we are learning to anticipate genetic errors and intercept them before they become irreversible.